Okay, everybody, I'm coming to you live from Kampala, Uganda, and we just had 8 more cases announced last night at midnight of COVID-19. So slightly distracted, but I'm going to try to tell you about what we're doing, and really happy to connect with people about the work that we're doing in East Africa for farmers. I'm presenting this on behalf of our team, which is the Cassava Virus Action Project, and you guys can find more information about that online at our website. And let me just, okay, here we go. Next slide. Okay, so before I start, I just want to make sure that I recognize the team that I work with. So this is a picture of the scientists that have been really crucial in starting this project of taking portable genomics and computing to farmers. We're an international team. On the far left is Monica Kehoe. She's in Australia. Then you have Dr. Joseph Nguru. He's a Tanzanian virologist. Then Dr. Titus Alachai is a virologist. Dr. Peter Serwaji is a Ugandan entomologist. That's me looking ridiculous. And then we have Professor Elijah Ateka from Kenya. And all of the work that I'm going to present today is a team effort. And I'm just the lucky one that gets to sit here and talk to you about it. We also have, okay, I think I missed one. What happened? Okay, so I'm going to tell you a little bit about cassava. This particular plant, the leaves and the roots are eaten by 800 million people globally, and 500 million people rely on this in sub-Saharan Africa alone. And many of you, this is at the time where if I were there, I would ask you how many of you know about cassava. And you would raise your hand and you would say, maybe I do and maybe I don't. But the biggest cassava producing country in the world is Nigeria, followed by Thailand. So many of you have actually seen or eaten cassava because it makes up the main ingredient in tapioca pudding and is also the small balls in bubble tea. But for many, in sub-Saharan Africa, it's a staple food. If this farmer, if she has enough cassava, she can feed her family and she can sell it at the market like you see here, and she can generate income for really important things for her family like school fees, medical expenses, and savings. And as I said before, 800 million people rely on this for their daily calories. And it's currently, so healthy cassava will give a farmer 35 to 40 tons per hectare. So here's a picture of cassava that is very healthy, and this particular farmer would be able to get a decent yield. But the current situation in East Africa is a lot of farmers are getting cassava that looks like this. It's unhealthy. And they'll usually get about 1 ton per hectare versus the 35 to 40. And so scientists in the region since probably the early 90s have been trying to figure out what in the world is happening to these particular plants. And you guys, there's a slight delay when I click the slide. I actually can't see the slide. Okay, so the problem that from 35 to 1 ton per hectare, what is happening to these plants? Whiteflies that you see there on the left are transmitting 2 deadly viruses. So on the top you see there's a disease that's caused by cassava brown streak virus and Uganda cassava brown streak virus, and that is those particular viruses are RNA viruses. And then also cassava mosaic disease is caused by about 7 or 8 different DNA viruses. So basically you have whiteflies flying around like little dirty hypodermic needles, transmitting these viruses from plant to plant to plant. And so that's what's causing this huge reduction in yield. And so one of the problem that we wanted to tackle with this particular project that we're working on is identifying what strain of virus is circulating in a field. When we started the project, the scientists in the region were having this struggle with sending samples overseas. It would take like 3 months to get a genetic sequence back. And we knew at the time that this is a solvable problem, right? We knew that these portable genomic technologies and computing solutions were being used in the fight for Ebola. And actually, this technology is on the front line now with COVID-19. So we knew that this new technology would be useful in solving this problem of scientists having to wait to tell farmers what viruses they have. And it's not just about giving them the genomic sequence, right? It's about giving them the solution. What variety of cassava is tolerant or resistant to the actual strain that you have in your field? And so in 2017, we got together and we decided that we would work as a group to bring together this collective of people. And we thought, let's travel to Tanzania, Uganda, and Kenya, and let's develop DNA sequencing protocols in the region. So not just, at the time I was in Australia, not just develop everything in Australia and then try to adapt it to the East African context, but we would work as a group to develop our protocols that were that would work here in East Africa. And so, as I mentioned before, the most important part of this was taking the lab and the computational resources that normally are locked up in the ivory tower, taking all of that out to the farm. And I have to say, the majority of people thought that we were absolutely insane in 2017. Everybody told us that we would not be able to do this. And our reaction was, well, there's no other choice. So we're going to give it a go. And so I wanted to make sure that a lot of you might not be super familiar with this new sequencing technology. So we heard this morning about the microbiome and all of the genomic sequencing, and now there's new technology where you have a handheld sequencer. And I put this slide up there. I'm not an expert on nanopore sequencing, but basically there are pores, there's motor proteins, and DNA flows through these pores. And as we were told, and we all know, there's A, T, G, and C in genomic sequencing. And each one of those base pairs gives a different signal when it goes through that pore. So one of the interesting computational challenges in this particular type of data is going from basically a squiggle or a signal to actually A, T, G, and C. So you can see in this slide, you have a pore, you have some DNA that's being made single-stranded, and then on the most important thing I think for this particular crowd is you'll see on the underneath the word algorithm, you have the signal, right? That's the raw data that comes through there. And so there's really fascinating computational methods that do they call it base calling, right? So that's one of the computational challenges that had to be overcome in the field, was getting base calling or the signal into the actual genetic data for that to happen as real time as possible. And so this is the technology, and I've explained a little bit about the analysis that happens. And so our first plan, so we rolled this out in several stages. So in 2017, we wanted to visit a farmer. We wanted to collect healthy and infected looking leaves. We extracted the DNA. We did a rapid kit. We did local base calling. And then we blasted the reads against a local database of known viruses. And then we went back to the farmer. So our farmers that we were working with in Tanzania, There's an amazing farmer named Asha. Here in Uganda, we worked with a farmer named Naomi. And in Kenya, we were working with a farmer named Rose. And I'm just going to highlight Asha because she is a typical smallholder farmer in East Africa. So we're talking about she has probably a couple of maybe one acre of land. This is her in the field with us in 2017, and you can see she's helping us collect whiteflies, and she's getting her cassava yield in 2017 was 1 ton per hectare, but we knew she could get 35 to 40, right? And so she was the first person that we started this portable sequencing for farmers initiative. And one of the things that's interesting about this is if you look at this particular picture, the only impact that our team is really interested in is more yield for these farmers, right? And so we need to be able to give them advice on what to do with this. And so you'll see on the left, the plant looks healthy. And on the right, the plant clearly doesn't look healthy. But what advice do you give this farmer? Do you tell her to just remove the plant that doesn't look good? Or do you tell her to remove everything, right? And so what we did at this time was we walked around with Asha and we collected samples. And you can see that the question of is that plant infected is an important one. So we took samples from both of these, and we went back to the lab. So at this point in 2017, we weren't doing everything on the farm because we didn't have portable DNA extraction at the time. And also computationally, we needed a computer that do the base calling. And at the time, there wasn't anything that would do it in the field. And in the process of doing this, one of the most important things that the team was interested in is capacity building of being able to do all the sequencing locally by the teams in East Africa. So this is the amazing team in Tanzania. And I just have to give a huge shout out to Dr. Joseph Nguru, who's there in the yellow lab coat, because he really truly is a visionary in spearheading he and I's collaboration and this whole regional team of cassava disease diagnostics. He's an amazing, amazing scientist, and he deserves a huge amount of credit for this. And so we spent a lot of time doing the lab work in their labs that they have in Tanzania. And we also did this so here's the team in Tanzania doing the library preparation. So we've taken the samples from Asha, we're back to the lab, and we also did this in Uganda. Oh my gosh, this is one of my favorite photos. You guys, I can't even interact with you. This is so bizarre. But anyway, this particular picture makes me happy. I don't know how many of you are familiar with doing library preparations, but this is the first time the team is seeing when you use magnetic beads and you're separating with the magnetic rack. So this was a big day for us to actually be able to prepare the library locally, because what typically happens is the DNA extraction or the RNA extraction will happen in the lab, and then the samples have to be packaged up and sent overseas for the sequencing. So this was an exciting time for us to do sequencing in the lab here in Uganda. And then also we've done a lot of really cool work in Tanzania with doing capacity building of teams at Jomo Kenyatta and also at Eldoret University. So this is part of our team. We have really two missions. One, we care about farmers, and two, we care about capacity building of scientists in the region. Okay, so this is a photo of our loading at the time for Asha samples. And I just want to point out that computationally at the time in 2017, we were using the Mac Pro that you see back there. And so we had to drive back to the lab to do the DNA extraction. And we also had to use the Mac Pro to do the base calling. And that was that I hand carried that and we left it there. So at the time we were doing this, we were having to use a cell phone as a Wi-Fi hotspot because one of the biggest challenges in the region is having access to the internet. And so at the time, the Nanopore device have had to ping the company with a signal. So we use the phone there as the Wi-Fi hotspot. And the Mac Pro we were using to do the base calling and the blasting. And blasting was happening via, we have a few terabytes on the right there, the silver machine, a hard drive, that we had downloaded some of the databases because we can't rely on the internet. It's not here. We have to have local solutions to do this. And so this was our setup that we used in 2017. And I mean, I had to show some data, guys, because this is a supercomputing conference. But I'm going to send you a link to the paper. But one of the main take homes of this is that plant that here's Asha's plant and the disease severity, this top purple one says healthy. And what's really important about that is it wasn't healthy. It wasn't a healthy plant, and the PCR result was negative. So it wasn't developed enough. So one of the things that we found and I think we would probably have to do a lot more sampling, but it seems like the Nanopore device is much more sensitive to infections, especially these viral diseases in plants. And so one great thing was that we're able to tell her, look, you need to get rid of everything here. It might look healthy, the plant might look healthy, but it's actually infected. And that particular plant we took back to the lab and we put it in the greenhouse, and it did develop full-blown symptoms. So we were really happy that our sequencing results were confirmed later. And another thing I want to point out on this slide is that in 33 seconds, we have a whole genome. So in 33 seconds, we have that the plant is infected. And it's not just it's infected or not, right? This is a matter of what strain is it infected with. So you can see there's different strains over here. And that matters because cassava breeders have been working on solutions against particular strains. And so if you guys want to look at that paper, we made sure that one was published. And okay, so just specifically about that particular study, we were able to give the results in 33 seconds. Honestly, when the PCR failed, we still got a positive, and there was no enhancement step for that. It was whole genome sequencing, right? We just ground up a cassava leaf and found what we needed. Okay. So in 2017, we went back out to Asha, and at that time, it was taking us 48 hours to do this. And this is the team. This is Charles Kayuki showing Asha, showing the farmer what we did with her sample, because at that time we really wanted to show her we went sample all the way to the solution. And so we told her at that moment, this is what virus, and then the team in Tanzania advised her to burn her field, which she did, and she planted the variety that was tolerant to the viruses that we found. And then 9 months later, we came back and she had enough cassava for the entire village. And at this moment, it was when I realized that we were doing something real and that it really mattered. And so I want to show you a video of her.
Imekoswakoswa na nyoka yule mwenye sumu kali, ati yeye na kapu lake anachukua anaweka wa
See the results right now.
Yes.
Asha has too much cassava. And that's a really great problem to have.
Na kuzalisha mazao mengine. Unaweza ukategemea ukauza muhogo, ukaenda kununua mchicha, ukanunua mbegu ya maboga, ukanunua mbolea, ukaanzisha bustani.
Good quality DNA. And okay, so I wanted to make sure that you saw firsthand what it's like, not just me talking. So the next bit, you guys, I don't even know how much time I have. I thought that thing said 2 minutes, but I think it's recording for 2 hours, so I might be going too long. But anyway, somebody scream at me if I'm too long. Okay, so our next mission in this, and this will be a lot faster than that, is we wanted to eliminate the time, right? We were now down to 48 hours, but we wanted to not have to go back to the lab, and we wanted to not have to be reliant on power and internet. And so our goal in 2018, what we're continuing to do now is to get the sample, get the answer while the farmer waited. And we called at this particular adventure, we called it Tree Lab because we wanted to take everything to the farm and we wanted to sit under a tree on the farm. So we set out to do Tree Lab in Tanzania, Uganda, Kenya, and we did this in 3 different places, 3 weeks time. And this is what the setup looked like. So we basically took everything that was needed from the lab, and that included some breakthroughs on computational resources as well. So, and I'm going to show you what that was. But the key here was we had Professor Jo Stanton from the University of Otago in New Zealand. Her team was independently working on a device that would extract DNA within about 30 minutes. And this was happening, and we could run the device off of a motorcycle battery. So I went and gave a seminar there, and she happened to sign up to talk to me. And I just said, hey, do you want to come hang out with us in East Africa? And she's like, yeah, I'll bring my machine. Cool. So you'll see her there in the burgundy shirt. And so she was testing her machine to do the DNA extraction real time. And then we were really fortunate that Oxford Nanopore gave us the first prototype of this device called a Minute, which basically they had been working with NVIDIA, and they put in a really fast processor to do that base calling that I was talking about. And so we set off to do this. And so here's the equipment that we had. This is all of our this is the molecular lab and the Minute, which you see there. We call that the mini supercomputer. That has the NVIDIA GPU chips in it, and that does the base calling real time. And so this breakthrough of doing the DNA extraction in the field all the way through to having the base calling happen was really crucial for us at this time. So in the top, you'll see the motorcycle battery that's running the DNA extraction machine, and then the little mini supercomputer and our sequencer, which is there in the silver, is being run by a laptop power bank. So that was all the equipment that we used to go from an infected cassava leaf to the answer in a short amount of time. So I just want to show some cool photos of the team doing some of the work. So this is Charles Kayuki doing the library preparation, and this is Charles loading the MinION flow cell, And it was cool because the women gave us the drum. We didn't really think, so that was great. We had a drum to load the first one, and this is just sort of the minute doing the base calling and our Minion doing the sequencing and being able to run it from a power bank. And so this is 3 hours later, we were able to give the farmers the results. And I mean, one of the things that here in Uganda doing now, I've moved here to work on this project full time. I mean, our outreach, we didn't really know it was going to work. And when it did, the farmers are holding my laptop. I mean, we're going to work on our extension and how to actually integrate these results into a platform that is accessible and scalable to more farmers. But this is us celebrating that it worked. We were surprised ourselves. And we then repeated this in Uganda. So this is the team in Uganda, and everything that we need to do this work is being carried by the team, which I think it's a massive breakthrough for all sorts of disease diagnostics. And this is the Ugandan team doing the DNA extractions, and we're loading the flow cells. And I mean, every time we get it to work, even at all, we celebrate. So that's us celebrating. And then this is Kenya. This is Brenda Muga. She's an amazing scientist. And this farmer is named Mama Mina. And Mama Mina, I mean, you guys, molecular biology is really not that exciting, to tell you the truth. And she is bored out of her mind, but she's also really happy to see us processing her samples, right? Because what normally happens is scientists go to a farm, take a sample, and then they never come back, or they come back months later. So the farmers, as much as this is not really that exciting, they're also very intrigued to watch and see what's happening. And they're most excited when we tell them the results of what virus they have or not, and what they need to do with it. And so this is the setup. I mean, I should have mentioned also that you visualize what's going on the little mini supercomputer by connecting to it via Wi-Fi with a laptop or a tablet or a phone or something like that. But this is I feel quite good about this, because in the end, we started to get neater on our little blanket. So I'm glad somebody captured us not in complete chaos doing something. So that's us in Kenya. We are generating the data real time, and it's being analyzed real time. And so really the take home message of this entire thing is it used to take up to 6 months for a farmer to find out what was going on. And by then, really, it's too late. The crop is already infected. And so we're really pleased and happy that we've now taken that down to a 3-hour time frame, and we can do it while they wait. And then that also means that they can take action against what's going on in their field real time. And so the big plans, which is, I mean, I just pieced out of academia, and now I'm doing this project all the time. After seeing real impact, I realized that this is a really cool moment in time to take the science to the people, and the people that matter the most. And so we're going to do this for more farmers. We also, the build capacity to do this helps with other crops and livestock. And also, it would be amazing if the teams in the region here could be ready to do sequencing of, obviously, COVID-19. So we want to scale the work that we're doing for farmers, like with mobile apps. So we're working with some app developers that are already hooked into giving farmers information like cost of cassava at the market and rains and all that. So we're going to try to incorporate these results in with those existing things. And really, the only thing that matters here is more resources for these smallholder farmers, because in our team's opinion, it's ridiculous that farmers in 2020 are going hungry. And we have the technology to do it, but the resources are just not evenly distributed around the globe. And so we're here hustling to make it happen. And I think I have a few more slides. Okay, so if you want more information and you want to see me a lot more polished, you can watch the TED Talk I gave, which nearly killed me, but I did it. And we also have, our paper has been published on this if you want to see more specifics. I mean, I know I didn't go into amazing detail about the analysis, but you can ask me questions or you can look in there. Really, honestly, like I'm really happy to have been given a chance to talk to you guys in the internet world, wherever you are, because HPC stuff requires a lot of power to make it work, right? So we feel like we do this. We have two lines of analysis. We have the in the field thing where we have to have all of our databases locally, and we have to have algorithms that are really smart. And then once we get back to a place with power and internet and access to enough bandwidth to upload data to an HPC facility, that's a whole nother set of a pipeline that we use to verify what we did in the field. So we're more than happy to work with people that have offline solutions or ways of making this easier, and because the HPC facilities are here, it's that last mile, right? Have some places in Uganda have zero coverage, nothing, zero. So anyway, would love to hear people's ideas about that. And I think I have my acknowledgement slide here that I want to say, honestly, our team we call ourselves a collective, and it's just spread out all over the world. And it really takes a big team to make things that haven't been done before happen. And so I'm personally grateful for all of these people. And I also want to make sure I thank the organizers of this conference. I have to tell you that with all of this stress and everything, I was like, I don't think I can do this. It's too much. But I have to say, over the last couple of days, I've really enjoyed the distraction of listening to all of you and reading the questions and everything. So thank you to the organizers who have put this together. And I too would love to come to Poland and visit with everyone. So with that, I'm just going to click to the next slide, which is where you can find more information about us. And I'm happy to take any questions.
Laura, it is wonderful work that you are describing.
You guys, I don't know where I start on this. Aw, thanks people for saying nice things.
Laura, one thing, the name of the conference is Supercomputing Frontiers.
Yeah.
I want to remind everybody that supercomputers of today will be implanted in your artificial teeth as your personal assistant in about 10 to 15 years.
Yeah.
So your Apple computer was a supercomputer about 20 years ago. So we're talking the same thing. So now there's the time for questions.
Okay, so one asked, what does it take to prepare a probe for that MinION device? Okay, so it takes us about, so as I said, you collect the leaf material, and then it will take about 30 minutes to get the DNA or the RNA, the genetic material, out of there. So that's 30 minutes. And then there's a kit that it's a, pipette, some clear liquids, and that takes about 20 minutes. And then that is loaded onto that flow cell. So, I mean, I think it took 3 hours to get an answer, but I think a lot of that was, you're labeling the tubes, and then you're trying, there's a lot of coordination of people. But I mean, I think that's going to get easier as well, because there's pipetting, which is always, I mean, farmers stare at us when we've got gloves on in the field. We just look ridiculous. But yeah. And you do post-processing a PC then at the farm? Yeah, so I think we do like I said, if you're going to collect cassava leaves, we download onto maybe an SSD drive or something all the cassava pathogens that are known. And because there's no capacity to use the internet to like blast, you know what I mean? We don't have any access to anything. So we have to bring the data closer to the farmer. So that's one of the things that I think we can get better at, because eventually this needs to be bigger than just cassava and you know, beans and sweet potatoes and maize and all of that. So I think there's lots of cool ways to make push the data closer to the problem. But with that being said, whatever answer we get on the farm from our local database, we always want to verify it using. I've been using the Pawsey Supercomputer Center in Australia when I was there to do the blasting and the post processing of the data, which is why the HPC thing comes in. What were the biggest challenges during sample isolation and data collection in the field? I mean, the whole process, I think one of the biggest challenges is the fact that we know there's no backup plan, right? We went really far away from anything that could save us, right? So I think that our biggest challenge and our biggest strength was that we were really organized as a team. We knew exactly who was going to do the DNA extraction, exactly who was going to work on the library prep. We were very coordinated in what we were going to do. So the whole process is a challenge, honestly. The whole thing was big preparation to make sure, because there's no, oh, I'm just going to run down to my collaborator's lab and get something. We had to have everything with us in the car. Do you think I have any more questions? Or I thank you guys for writing some. Okay, maybe we have something about HIV here. Your lecture did already answer part of the question. Is that technology applicable to other viruses beyond plant viruses, etc.? Yes. So technology is actively being used for pretty much everything, right? We're pulling out the viruses, the viral reads, but you know, like I had mentioned, it would be worth, if people are interested in the COVID-19 rapid sequencing thing, there's a bunch of really amazing scientists who are leading the way with this technology to get whole genomes of the viruses. And also people use the technology for plant genomes. So one of the advantages of this particular technology is it's long read data. And the technology is very disruptive. So of course, when it came out, there were lots of criticisms about all sorts of things. One of the things that people criticize it the most for is like accuracy. But through algorithm development and different pores in the brain, the accuracy has just skyrocketed through the roof, and it's equivalent or superior to some of the older sequencing technologies. So yeah, that's my thoughts on that. I don't even know if I answered the question, you guys, seriously. You guys, thanks for saying nice things. Okay. Yeah, you guys, I should have said this. 80% of the farmers in East Africa are women. And so, yeah, and so the last question here is, all of the farmers were female. Does that reflect the actual profile of a farmer? Is that just women who are more willing to cooperate? And so, as I just said, 80% of the farmers are women. And one of the things that I've found, a lot of people will say to me, and I think we're figuring this out as we go, is how do you scale the results? And the thing is, African women farmers are scaled already, right? That we met Asha, but Asha was riding deep with all of her women there, right? And then they have one dude on the team, and he kind of, you know, he's there doing whatever. But these women work farm by farm. They work as a collective. And so for us giving the information to Asha, when we went back 9 months later, she had actually engaged with the entire village. All of them were involved in this. And so The short answer is the majority of them are women. Okay, now there's something here about, you mentioned that there was only one read for one PCR negative sample, but samples were barcoded and pooled. Rapid barcoding procedure may cause sample crosstalk. Did you? Yes. Okay. So the sample that was negative, or what we got a viral read, we took that plant back to the lab, and then over time it developed symptoms and it was positive for a PCR. And so we felt pretty good about the fact that we had told her to remove the things that looked healthy.